The difference between mitosis and meiosis is that mitosis is the basis of growth, cell replacements, and tissue repairs. As for meiosis, it is the basis of sexual reproduction with gametes, such as egg, sperm, and pollen. The cell cycle starts when a new cell forms and runs through interphase, which is the longest phase. Interphase consists of 3 stages. The first stage is G1. This is a stage where a cell grows in mass, doubles the number of cytoplasmic components such as Golgi body and ER. The second phase is S. This is when the DNA duplicates chromosomes. The second stage is G2. This is the final prep before the cell starts dividing and goes into mitosis. Prophase is where duplicated chromosomes start to condense. This is also the phase when the nuclear envelope starts to break apart. Metaphase is when the chromosomes line up in the middle between the 2 spindle pools. Next step is anaphase. This phase is when the sister chromatids detach from each other. Phase. One chromosome of each type reach the opposite spindle pools. This is a phase when the nuclear envelope comes back and forms around each cluster. Lastly, we have cytokinesis, which is the division of the cytoplasm. Cytokinesis is not considered a part of mitosis, however, it is the last step of the cell cycle. All chromosomes are duplicated during interphase before meiosis. Meiosis consists of 2 divisions, meiosis I and meiosis II. During meiosis, one cell divides twice to form 4 daughter cells. The first step of meiosis I is prophase I. When chromosomes condense and align with their homologous pair. Each pair goes through crossing over, which breaks off DNA and swaps, and this is also when the nuclear envelope breaks up. Next we have metaphase I. This is where the homologous chromosome pairs align randomly at the spindle equator. Anaphase I is next. This is where microtubules separate each chromosome from its pair, and as anaphase I ends, one set of duplicated chromosomes near each spindle pole. Lastly, we have telophase I and cytokinesis. This is where the 2 nuclei start to form and the cytoplasm divides. Starting off with meiosis II, the next phase is prophase II. Now that you have 2 daughter cells, the membrane around the nucleus of each daughter cell dissolves and the centrioles duplicate. After prophase II is metaphase II. The pair of sister chromatids line up end to end along the equator of the cell. Spindle fibers at each pole of the cell attach to each of the sister chromatids. After that is anaphase II. The sister chromatids are pulled to the opposite poles, and they are now separated and are individual chromosomes. Lastly, we have telophase II and cytokinesis. A new nuclear envelope encloses each parcel of chromosomes, so as a result, there are 4 nuclei or haploid cells. Now we're going to talk about the cytoplasmic division in animals and plants. Starting off with animals, in animal cells, a contractile ring contracts and pulls the cell surface inward until the cytoplasm is divided into 2. Now for plants, it's a different story. In plant cells, a cell plate will form. The cell plate becomes a cross wall that partitions the cytoplasm and divides it into 2.